Okay, good morning, everyone. Today, our speaker is Dr. Dave Zhao from University of Illinois at Urbana-Champaign. And Dr. Zhao is an associate professor in the Department of Statistics there. His research interests include shrinkage estimation, multiple testing, integrative genomics, and spatial transcriptomics. Dr. Zhao received his bachelor's in chemistry and physics and the doctoral degree in biostatistics from Harvard University before completing a postdoctoral fellowship in biostatistics and statistics at University of Pennsylvania. And today he's going to give us a talk about his research on mediation analysis. Let's welcome Dr. Zhao.
Thanks a lot. Thanks for the invitation. Although I have to say that when I looked at who else spoke in this seminar series this year, I was very intimidated. So I hope that this, is somewhat interesting to everyone. Yes, so I'm going to talk about mediation. I was going to present some of the stuff that we've done in a narrative format, kind of. And I wanted to tell you first how I got into this. This area, and it wasn't direct really. So it started when I was a postdoc, and I was working with a group looking at heart failure at Penn, and they had so they were doing genomics research, and they had a data set where they had non-failing and failing patients. For each patient, they could measure their DNA, their genotype, and they also had information for a lot of different genes about how highly those genes were expressed. So they did their study and that was fine, but then they kept asking me, hey, we have these 3 different types of data. We have their outcome, we have their gene expression, we have their genotype. And it seems like we should be able to use them all together in some way. Seems like we should be, for example, a standard analysis is, can you associate the outcome, failing or non-failing heart failure, with the genotype? That's a sort of genome-wide association study. It's a pretty common thing that people do in genomics. But they said, hey, we have this other type of data. Shouldn't we be able to do better? So that was sort of the question that they posed. And it wasn't really one that I was able to answer during working with them, but it kind of stuck in my head for a while. So the main question that I started looking at was, suppose you do have these 3 types of information. Can you use this gene expression information to get more power for this association analysis? So then I was like, okay, let me try just the simplest thing I can imagine to combine these data types together. And the simplest thing stems from the central dogma of molecular biology. And it says that information goes from your genetic code through gene expression to everything else, proteins and how things work and so on. And so I thought, okay, well, I don't really know why this would work, but let me just pretend like I'm going to model my regression models based on the central dogma, and I'll just see what happens. So I ended up trying to write down this model, and S here stands for the single nucleotide polymorphism. So this is the genotype data. G here stands for the gene expression, and Y here stands for the outcome, so heart failure or no heart failure. For now, let's pretend like it's continuous. And so according to the central dogma, the most distal phenotype outcome is a product of the gene expression levels. And the gene expression levels themselves are a product of the genotype, of the SNPs. So that's what this equation, or these 2 equations say. And then the ultimate association I'm interested in is between S and Y. And so if you just substitute in this equation into this equation, then you find that this parameter, beta 0, which is the product of this capital B 0 and alpha 0, that's ultimately the parameter I'm interested in. So beta 0 quantifies relationship between S and Y. Does that make sense? So the goal is to do inference on beta 0. And the question is, if I write down this, can I do better than if I just regress Y on S, which should just give me beta 0? Standard, regression, usually what people would do in a genome-wide association study. So, okay, so I thought, okay, well, let's look through this. So let's look at the OLS estimator. What kind of distribution does the OLS estimator have? And you can write down the usual expression. You substitute in the expression for y, and then you substitute in g into here, and you can work things out, and you find out that this is going to be the distribution of the OLS estimator. Okay, great. Now let's see what happens if I use this extra information g. So intuitively, what I thought you should do is, okay, well, I have this extra information. Let me just regress y on g first, get an estimate for alpha. Then g times my estimate of alpha should roughly capture the information about my phenotype, my outcome. And so then I'm going to regress g times my estimate of alpha on my s. And so I do that. And if you work through sort of what happens here, this A hat is my estimator for alpha 0. Assume that everything's nice and easy. Let's assume there's a single G and a single S. This becomes my OLS estimator. I expand Y in terms of this model, these two models here. And then I actually get a different distribution for B hat. So I looked at this and I said, well, what is this? And if you stare at this part over here, if you only have a single G, you only have a single S, and everything's centered, then this is basically the squared correlation between S and G. So we had the covariance here, so squared covariance divided by variance of G divided by variance of S. So that gives me a squared correlation, which says that this part always will be less than this part, which is wild. So basically, this wonky estimator that I just dreamed up has apparently more power than OLS. So the eventual answer to their question, can you gain power, appears to be yes. If you have this kind of multi-stage phenomenon and your model is correctly specified, it looks like you can gain power. So I looked around for what this phenomenon even was. And I wasn't familiar with mediation analysis before working on that project, but mediation analysis seemed to be the best framework to describe what I had just done. I looked at a couple other things like two-stage least squares and measurement error models and things like that to try to frame that calculation that I had just seen, that you had just seen. And mediation analysis seemed to be the best one. So in the language of mediation analysis, I had an exposure, which is my genotype, and I was interested in decomposing the effect of my exposure on my outcome, which is my heart failure. And that effect can be broken down into either a direct effect, where you can imagine some direct causal relationship, or an indirect effect, where the effect of the exposure first affects some intervening variables. And in my case, those were my gene expression levels. So this is what brought me to mediation analysis. I sort of got here from a genomics consideration. The rest of the talk will basically be about this triangle here, trying to figure out under various conditions, for example, high dimensional G, whether you can estimate the indirect effect or the direct effect, how to get valid asymptotic distributions on your estimates, and some discussion about this power issue that I just described. So that's sort of how I got here to mediation analysis. So that first work eventually became this paper, and we worked it out for linear models, logistic models, generalized linear models. And it looked in simulations and in the data analysis that you could in fact gain power. So then I started digging into the mediation literature because I couldn't imagine that other people hadn't realized this. And so the story there is interesting and still one that I'm trying to piece together. So in 2014, which is when our paper was published, the only paper that I could find about this phenomenon was this one. And yeah, so this phenomenon appears to be really discussed a lot in psychology. And so this paper had a simulation study that suggested some kind of power anomaly. And in particular, the one that I described. They said that if you test for the indirect effect, so in this picture, if you test only for the blue in the absence of an orange, that has higher power than just testing Y on S, which is exactly what I described. And so they found that in simulations, and I just couldn't find anybody who had worked out the math. Since then, this paper was in 2015, and they warned that, yes, you can have power. They work out the math, and they say, beware, this requires a lot of assumptions. And then in 2018, this paper works out a little bit more of the math and looks at, I think, other properties of the estimators and so on. So I realized that actually what I had done fell into, I think, quite a bit of literature in the mediation world. It turns out that the estimator that I described, if you have a linear outcome, is the product method, the standard product method from Barron and Kenny. And it's very well known. I didn't realize the equivalence at the time, but that's a very well-known estimator. So this sort of got me into this field. I started reading a little bit more about mediation and trying to see what others have been doing. And still, to the best of my knowledge, this power phenomenon doesn't seem to be that well described outside of the psychometric psychology literature. So since then, I've been trying to figure out two things. And the first thing is, we have this cool power idea. How can I make it more useful in practice? Everything that I showed you assumes pretty simple situations, low dimensional G, low dimensional S, and nice linear models and so on. But in real life, if I really wanted to give this to these heart failure genomics people, I'd be a little bit hesitant because it's not quite realistic, I think. And the second question I've been thinking about is, what exactly is going on with this power situation? I can show it, I can show the math, I can show it in simulations, but I don't really get it. And so at the end of the talk, I think there are three different ways that I think about what is actually happening, and I haven't quite pieced together the full story. Does that make sense? Any questions so far? Also, I should say that when people give talks in my department, I interrupt a lot because I just am interested. And so if you ever want to interrupt or discuss something, please just turn your mic on.
Yes. So I have a question. So it seems like there's something important missing from your model, and that's the environment that gene expression depends on signals received by the cell. And right. So is that something that we're just leaving off for now so we can work out the model and we're going to add in later?
Yes. The short answer is yes. I haven't figured out, that's a very important part of the model that's missing. Another important part of the model that's missing is confounders. So these arrows may not go the right way. And for now, yes, I'm leaving them off. I haven't really figured out how to handle them. I mean, you can always put them in, you can always write them down. I just don't know whether this power thing still carries over. And yes, that's definitely something that I want to think about in the future. I should say another issue here, which I'm sure you also have guessed, is that this arrow appears between G and Y, but this assumes that all of these genes are causal for the outcome. Of course, some genes can be reactive to the outcome, so you have heart failure, and that induces some kind of response in some other set of genes. I don't know how to handle that either. The result that I just showed you is highly, highly dependent on this very simple situation here.
Gotcha.
Yeah. I mean, that's a great question, and yeah, one of the things I want to think about.
Right. Also, another question.
Yeah, go ahead.
Yes.
Okay. So in the case whereby, like according to this graph that you're showing here, this triangle, in the case whereby the direct effect is probably very minimal, while the indirect effect is very large, like for example, the mediator has just that high impact on the outcome. What will be the difference between this mediation analysis and the instrument variable analysis?
Yes, okay, so great question. First, I want to emphasize here that in this work, I don't have a direct effect. So I am indeed in that exact situation that you described.
So this is technically instrument analysis. instrument variable analysis.
Yes, and I think the only difference, I mean, the biggest difference is instrumental variable analysis is interested in this arrow here, right? It's interested in understanding this effect. But I think I'm interested in understanding this overall S to Y effect.
Okay.
And that I think they're very closely related. To the best of my understanding, instrumental variables tries to tease apart the causal nature of this arrow here, whereas I'm interested in pure association between S and Y.
Okay.
Yeah.
Yeah, so Dave, I have a related question. I think even that, because this is essentially a causal graph, right? You're assuming a causal relationship between S pointing to G, G pointing to Y. Then, given that we assume this causal relationship and we can estimate each causal effect, then why would we still interested in the overall association between S and Y?
So you're saying if I can get each of these separately?
Exactly, yeah.
I guess I would say that this is that's sort of the exact point of what I am showing you. If you can get them separately and you use them to test overall, then you can do better than if you just tested overall. So yes, if you get them separately, sometimes you do want to, let's say, quantify how associated S and Y are, and you would quantify that overall association using the separate causal effects. And the whole point is, if you do that, you can do better than if you didn't have G at all.
Right. So I guess maybe I can provide the answer to my own question. I guess maybe the students will be interested in knowing why the overall association is interesting to the genetics field. Why do we want to use genotype to predict outcome or phenotype? I think the fundamental reason of doing that is because in clinical practices, it's only feasible for us to measure genotype from patients' blood. So if we can do that, yeah.
Yes. Okay. So practically speaking, why are we interested in the overall association? And yes, there are two reasons. First is if you want to do prediction, then feasibly measuring genotype is the easiest thing to do. But also in terms of understanding biology, there's a big interest in understanding the genetic architecture of certain diseases. And recently there was this paper that showed that if you really want to understand all of the genetics going into height, you need about 4 million people. And the reason for that appears to be that height is a highly pleiotropic condition where a lot of these S's have tiny effects. And if you really want to capture all of them, you have to have a huge sample size. It's possible that this method, once it's worked out, can help you reduce the sample size. So you still find information about the genetic architecture, which is this overall S to Y relationship, but you find it with more power using things like G. And I would say, yeah, there's a prediction reason as well as an inference reason why you want to do the overall effect.
And there's a comment from the chat box. Also, polygenic risk scores can guide behavior, like diet, exercise, etc. Yeah, that's a very nice comment from them.
And yeah, so this is getting ahead a lot, but polygenic risk scores are something that I'm interested in because this right now is about association, but presumably you could use this for prediction. So if you found a G that is relevant, you could potentially get more accurate beta hats, and you might get a more accurate prediction. And in the case of prediction, these arrows may not matter as much. The environmental effects, the confounders may not matter if I'm focused purely on prediction, for example, in polygenic risk scores. And so that's definitely something I want to look into as well.
Yeah.
So thanks for the comments. This is, yes, I mean, I think you're pointing out all the holes in this model, which I agree with and have not fixed and don't know how to fix yet.
Yeah, and also another thing I want to point out, but probably we can defer this to our Q&A, is that I think what you're discussing here is very related to what people call the TWAS right now, right? The transcriptome-wide association analysis, yeah. But I think what's special there is that their G, the mediator values are basically predicted.
Yes, and I think maybe we can talk about this later, but I think the purpose of TWAS is a little different still from what I'm describing.
Okay, yeah.
But,
okay.
Maybe we'll talk about that later.
I have a quick question.
Yes.
So I understand that you may have a power gain when you have a single S and a single G.
Yes.
But in GWAS, or we often have a million S and tens of thousands Gs.
Yes.
Is the power gain still there when you consider all the combinations?
Yes. So let me talk about the million G, 100,000s of Gs, but not million S. The answer to your question is yes, the power gain is still there under certain conditions if you have high dimensional G. The method that gets you there is harder, but we can't figure out how to get high dimensional S and high dimensional G to work. I don't remember why. However, as in standard GWAS, when people sort of do marginal association between Y and S, we can still do that here.
Okay, thank you.
Yeah. So great. So let me talk about this. So this was the obvious next step for us. So in low dimensions, we worked everything out. But right, in practice, you have a genome-wide, a transcriptome-wide assay, 10, 20,000 gene expression levels. Can you use all of them? So same model, same situation. It's just that G is a high dimensional vector, larger than sample size. Can you still gain power? So I'm going to take you through a bunch of things that we thought about that don't work. First, obviously, OLS doesn't work because you can't invert that G transpose G inverse, right? Second thing a lot of people have tried is some type of model selection or dimension reduction. So for example, do some type of screening on the gene expression levels, go from 20,000 down to something manageable, 10, 20, whatever. Other people have tried doing PCA, where you just find the principal components, project the gene expression down to the couple top principal components, and then you have a low dimensional situation there. And the issue there is that if you do screening, that doesn't take into account this post-model selection inference issue. If you do dimension reduction, the model is actually different from the one you wrote down. And so we weren't super happy with how those worked. The natural solution would be the lasso. If you have high dimensional G, then you first regress Y on G with a lasso instead of a OLS. And so I'm just calling that estimator A for now. And then you do a regression of G times a hat lasso on S. So this is just replacing this OLS estimator for the effect of Y on G with a lasso estimator for the effect of Y on G. So this doesn't work either because ultimately we're interested in inference, valid inference. And the asymptotic distribution of the regular lasso estimator is difficult to work with. So we passed on that. And then we thought about the d-bias lasso. And the way the d-bias lasso works is you take the original lasso estimator and then you subtract off some stuff or add on some stuff. This gives you, and I'll go over this maybe more in detail in a little bit, but this gives you an A hat which is asymptotic, which is a normal plus an OP1 error term. And then I should say that, so this is a high dimensional multivariate normal because the dimension of alpha 0 is p dimensional. So this OP1 is a vector, a p dimensional vector where each component is little OP1. So that actually turns out to be the problem with even the d-bias lasso approach. So the total effect that we're interested in, again, is the regression of my G times my d-bias lasso estimator on my S. If I write this out, I get a normal term, which is what I want, but then I have this term here. And this piece especially is the sum of P OP1s, little OP1s. So even if each of those components goes to 0 in probability, because P is high dimensional, I'm summing over an infinitely increasing number of OP1s, and that may not go to 0. And so I may not actually have an asymptotically normal distribution for my B hat, even if I use d-bias lasso.
So Dave, sorry for the interruption. I missed that part. So why wouldn't model selection work? So in the second line.
If you perform model selection and you do the appropriate post-model selection inference, then it would. But the existing techniques don't really do that. And post-model selection for this second stage mediation step is not something I've seen a whole lot yet.
I see.
Yeah.
Can I ask about the lasso?
Mhm.
So I think Ross Jacobucci and Safraz Sarang have written about this a little bit, where they recommend using the lasso for what they call exploratory mediation analysis. And they recommend using the lasso for variable selection, but then re-estimating using an OLS model in order to handle the bias in the estimates. What do you think about that approach?
So I'm not aware of that paper. And actually, if you wouldn't mind typing those names to the chat box, that'd be really helpful for me. But I have two comments. The first is that there was a paper by Zhang, and the last author was Liu at Wash U, and they used MCP instead of lasso to do this same idea. And we looked at that in simulations, and I don't actually have the simulations here, but the behavior was really odd. And basically the power was it was very high when the total effect, when the actual total effect was low, and the power curve actually went down as the total effect got larger. It was a really strange looking behavior. And we think it had to do with how even if you do that pre-screening step with a method that supposedly has the oracle property, in practice, if you don't properly account for that model selection variability, it still is an issue. At least in the simulations that we tried, that type of pre-screen and then fit didn't work very well. That being said, the second thing I have to say is that there's this paper recently that I haven't read by, I think, Witten and Shoje, and it says you can do the just pre-screen and refit. It's like, they're like in defense of this simple way of doing post-model selection inference. And I haven't looked at that, but it's possible that using those ideas, something like what you described might work. I'm not totally sure. So I guess in short, at least in simulations, this whole lasso or MCP plus refit didn't seem to work too well for us. So for the debiased lasso, we were also unable to do that. And the main issue, again, boiled down to this part right here. You can think about this as a contrast. So now I'm going to try to link this to some of the current work in the high-dimensional inference literature. This is not inference on a single component of a lasso estimator. This is inference on a contrast, a linear contrast of the lasso estimator. The problem is this G is a dense linear contrast. There's no guarantee that a lot of the entries of G are 0. If a lot of the entries of G were 0, if G were a sparse contrast, you have sparse times this thing, and then only few of the components of alpha tilde are going to matter, and we'll be fine. Because this is a dense contrast, you end up with this problem here where you have a lot of error terms being summed over each other. So that basically was a statistical issue that arises if you have high-dimensional G. So I'll try to give you an idea of what we did to resolve this issue. The high-level idea is that instead of using these debiasing methods for alpha 0, we applied this debiasing idea to this whole thing in the box. In other words, we're directly debiasing this dense contrast. I guess I forgot to write the other papers that do this, but it turns out that around the time we submitted this, a lot of other groups were looking at similar things. So there's a paper about approximate residual balancing in the causal inference literature from Athey and Vager and other people that do basically the same idea. So what I'm about to describe to you, I think, has been described several times in the literature. So what we're going to do is still consider an estimator of this form. So this looks kind of like what you would have with an OLS estimator, except instead of having the usual, G transpose G inverse G transpose, we're going to replace what we would usually put in OLS with this data-dependent matrix. And we're going to eventually figure out what matrix omega hat we need so that everything works out. So intuitively, we're just, again, going to start by considering this kind of estimator. And then if you expand y in terms of the two-stage model that I showed you before, with no direct effect, you can get this situation. So this is what y is equal to. And then you arrange some terms, add and subtract some things, and then beta 0 is equal to this right here. I think I'm missing an inverse. No, this is right. So you rearrange some things and you find that this term involves a high dimensional alpha 0. This term is going to be probably close to normal because this is an epsilon and this is sort of like a central limit theorem type of situation. And then this term also will probably turn out to be asymptotically normal as well. And so if you then are able to get rid of this, it looks like we can have an estimator that is centered at the true total effect that has asymptotically normal errors. So if this thing in the box is small, then we can have a good estimator. So it turns out this is not small, but we can estimate it well, because if we can estimate this using lasso, then we can try to get a good estimator of this, and then we can subtract this from both sides, and then we'll get a small term here. In other words, this is like a bias term. So this is where the debias word comes in. We don't have a good estimator here because it's biased for beta 0 by this amount. We're going to estimate that bias, subtract it from both sides, and then hope that that remainder is small. So we can do that. Basically, we estimate this bias by replacing alpha 0 with a lasso estimator. Now, if we can argue that this bias, this remainder term is small, then we have a normal error for our estimator. And so the reason this is small is because we can construct omega hat such that that first term involving omega hat is small. By construction, we can make it small. And then the second term involving alpha 0 minus alpha hat, that is small because of properties of the lasso. So lasso is an accurate estimator, and it is accurate enough such that even when we multiply it by this other term, that will go to 0. So if you rearrange that first line, sorry, this is just saying we need to construct omega hat that way. So if you rearrange the first line, you find that this is the estimator. It looks just like what you would expect if you blindly use the lasso, plus some term to handle this debiasing business. This omega hat is constructed using this constrained L1 minimization problem, where it is exactly constructed so that that term in the remainder is small. So this is similar to other work that's been used to estimate inverse covariance matrices. If you replace this term with the identity matrix, then you're basically looking for an omega that approximately inverts this covariance matrix for your G. In fact, if you replace this with the identity, that optimization problem is basically what is used in the standard debiased lasso methods. Once you replace it with a different term, this allows you to directly debias a high dimensional contrast. So that work ended up in this paper, and we were able to show a couple things. First, this omega hat will be close to some population level omega as the sample size increases. The second thing we were able to show is that our estimator is asymptotically normal, centered around the true total effect. And this term, this extra term is just like in the low dimensional situation. You can show that the OLS estimator has higher variability. So if this is a matrix, then this matrix minus this matrix is positive definite, or positive semi-definite. And then we had similar results for incomplete mediation, although not the power result. So here, for the first three things, I've assumed that S affects Y only through G. We also have results if you allow S to also directly affect Y, except not number 3. So the message here is, yes, in high dimensions, under these constraints, or linear model, everything's well specified, it'll still work. With this slightly different debiased lasso approach, where you debias a dense contrast instead of debiasing each component individually. Does that make sense? Okay, so this is just published, and this is about where we are now. So there are a couple things that we still don't fully understand. The first is, so I've talked about this power thing. I thought it was really neat, and it's such a cool property. But oh, I'm sorry. Let me just let me skip this actually. So there's this cool property, but what happens if you don't have complete mediation? If you have this extra effect between S and Y? So far we haven't allowed that. And so we started wondering what would happen if you allowed that. Do you still have this power gain? And this is some work we've been doing. So that previous work was with my student Rachel. And this is work that Rachel and I have been doing with David Zucker at Hebrew University in Jerusalem. And it's the same model, same situation, except we're adding this arrow back. If you add this arrow back, this term appears here. This didn't used to be there. And now we're still interested in the total effect. And we're now asking, if we allow this direct effect, can we still gain power? So there were a couple of things we tried. The short answer is not really, unless a direct effect is really small. That's sort of what we ended up discovering. The first thing we tried was this projection statistic idea. This is actually, and this is in low dimensions, but this is exactly what I described before in the situation where you don't have a direct effect. This is you regress Y on G, and then you regress the result on S. We thought that this may still work here. And the reason is that this first part kind of projects it projects Y onto G. So it contains the information in Y that can be contained in G, that can be captured by G. Then, because we have this assumed relationship between S and G, we hoped that that G information could then be regressed on S, and we could still gain power by using this intermediate, this mediating G. So if you write out what this test statistic actually equals, it is centered, or not, it has the true effect that we care about, total effect. It is normal, has normal errors, but it is not unbiased, and it has this term here. So this term causes some potential issues if we want to use this as a test statistic. So this is the direct effect. This is the indirect effect, beta S. And if we want to use this to test the total effect, then we better make sure that this is large if the total effect truly exists, right? So the total effect is beta S plus alpha S. And we hope that this is large if the total effect truly exists. So the problem is it may not be large because beta S and alpha S may have different signs. And so that's why we need to talk a little bit about some additional assumptions. So if you have a direct effect and you still want to use this as a test statistic, in order to maintain type 1 error, you need to assume that alpha and beta have the same sign. And that's because of what I just discussed. If the total effect exists, this better be large in magnitude. So in the mediation literature, this apparently is called consistent mediation. And this kind of says that the effect of S on G can't contradict the effect of G on Y. If you have that, then under the null hypothesis, this is the distribution of your test statistic, and you will still have type 1 error. For the power situation, we can't really analyze the power of this test statistic very well because of this extra term here. And so if we wanted to compare it to the OLS estimator, we had to use a local power kind of idea. where we assume that this alpha S goes to 0. It's very weak. If you have a weak direct effect, we can tell you when this projection test statistic is more powerful than OLS. It's when this situation happens. It's still kind of complicated, but basically, if the variance of if the residual variance of Y on G is large, this test statistic will have power. If the direct effect is small, this test statistic will have higher power. So basically, in general, we can't really gain power if you have a direct effect. If the direct effect is small, then we still may be able to, under certain conditions, where you have to have this consistent mediation to maintain type 1 error. And so we also worked this out for high dimensional G, and the story is basically similar. And if you look in simulations, this is borne out. This line here, this purple line here, is that test statistic I just showed you. Some of these other lines interpolate between that test statistic I showed you and the usual OLS estimator. We tried different sort of convex combinations to see if we could borrow strength sometimes and from one and borrow strength sometimes from the other. And it looks like if your direct effect is small, just using that original test statistic, the projected test statistic, has highest power. This green shaded area is the region where we have consistent mediation. If the direct effect is larger, then depending on the size of the relative size of the indirect effect, the total effect estimator may be better than what we're describing. So the story there is a little messier. I mean, the main thing is for this power thing to hold, you need a weak direct effect. And it was a little, I'm glad we found that out, but it was a little disappointing, I guess. So because of that work, we now have a better idea of what situation we need for this power phenomenon to arise. First, we need things to be really well specified. We can't, we have to have a really good reason to believe that those arrows look the way they look. And the second thing is we need probably this kind of consistent mediation where we need the signs to be the same. And so we realized actually after talking to one of my grad school friends that those conditions are usually assumed in this surrogate marker literature. So the last thing I'll talk about is this work with Rachel and Layla. Layla's at Rand, pretty close to you guys, and she works on surrogate markers. And the idea behind surrogate markers is if you have some sort of long-term outcome that you care about, but you have to wait a long time to measure it, or it's really expensive to measure, or it's not easy to measure, you might instead want to measure a surrogate. For example, I think Layla works on diabetes. So her long-term outcome is diabetes. Her interim outcome is, I think, blood glucose level, or one possible intermediate outcome is blood glucose level. So if you could capture a lot of the effect of the whatever disease or drug, drug here. If you want to capture the effect of the drug on the outcome, but you can't really measure the outcome, it'd be great if you could just look at the effect of the drug on the surrogate. And then that hopefully will be able to tell you a lot about what the true outcome effect would have been. So this is surrogate marker literature. These are, so this is Layla's earlier work on measuring just how much of that effect between the intervention and the outcome can be captured by the surrogate. And it turns out that for this surrogate literature, these people have also realized this power phenomenon. So if you look at this arrow structure, it is exactly like what I showed you in the very beginning. There's no direct effect here, or they don't put a direct effect here. They assume that the directions are exactly as we think they should be. And so then it really makes sense that if you want to test for the treatment effect of intervention on your clinical outcome, you might be able to get higher power if you use the surrogate as well. So we worked with Layla to study this problem, not the power problem, the proportion of effect that you can explain with surrogates if your surrogates are high dimensional. So here's the model. It's almost exactly like it was before. This is high dimensional. S now stands for surrogate, not SNP. E stands for exposure. And in this case, we're going to say that there could potentially be a direct effect between the, let's say, drug and the outcome. And we want to know what proportion of the total effect goes through the blue. If the proportion is high through the blue, then S is a good set of surrogates. And we can use those to do what I just described, test early, get more power, and that type of thing. The model here is a little different. Here, basically, we're assuming that the relationship between S and Y can differ, depending on whether you got a drug or whether you did not get a drug. And we're right now studying this problem. So given this model, high dimensional S, how can you estimate the proportion of effect going through blue? I don't think I have results to show you there. I mean, basically the same debiasing idea works out. You can get these asymptotically normal estimators for the proportion of the treatment effect, and roughly the same story holds. Okay, so the last thing I want to talk about is what exactly is going on with this power thing. And I don't really know the answer, but I have 3 possible explanations. So for now, let's just think about univariate Y, G, and S. If we care about the association, the correlation between Y and S, and we make no assumptions at all, then the Fréchet bound says that this must always be true. So if you just write out the correlation matrix between the 3-dimensional or 3x3 correlation matrix between Y, G, and S, and you use the fact that it has to be positive definite, then you can work out this relationship. So in general, knowing the correlation between Y and G and S may not tell you a whole lot about the total effect that you care about. Whereas what we have done in that first project that I showed you is basically assume this. We assume that if Y is correlated with G and G is correlated with S, we assume that Y must be correlated with S. That cuts this bound down by a lot. And so I think one reason for why we can gain power is we add a really strong assumption here. We looked at whether there were other ways to weaken this assumption that without going this far, and I don't think we ever came up with anything. But this is one possible explanation for where this power phenomenon is coming from. The second intuitive explanation for what's going on is denoising. So if you regress Y on S, if Y truly equals some effect of G plus error, then this error term is hurting you in terms of power. And if you could regress just G on S, or G alpha on S, then you wouldn't have this extra error term and you would get more accurate inference. And what's happening is if you don't collect G at all, if you don't collect data on G, all you can do is regress Y on S. If you collect data on G, you can try to estimate this G alpha. And it turns out that you can do it better than if you didn't have G at all. So you can roughly reduce the noise in your outcome. So, despite the fact that this is random, it turns out it still reduces noise compared to not doing anything at all. And the last thing that might be going on is based on this project with Layla. If you think now about a situation where you have only a binary exposure, so drug or no drug, and then in the drug group, in the treatment group, you have some distribution of your surrogate, and then Y is a function of your surrogate. And in your treatment group, it's the same situation. And now you can ask, what is the effect of the treatment? And the treatment effect can be written like this. And this shows how the effect of treatment is related to these functions F1 and F0. So this treatment effect is the analog of this total effect that we care about. You regress Y on your exposure, this direct total effect. And here it shows that if you know these F1 and F0, you can estimate that total effect. F1 and F0 are the associations between my mediator S and my outcome Y. And I don't know those associations and I have to estimate them, and that's where sort of the problem happens. If you make the assumption that this association, this function is the same between both treatment and control groups, what you can do is basically increase the sample size that you have to estimate those functions. So here in black, imagine black is the treatment group data points, and in circles, open circles, are the control group treatment points, are the control group points. So if you only had the black, then you couldn't estimate this curve very well. This curve is f of s. If you only had the white, you couldn't really estimate this curve very well. If you have both, and you assume that this curve is the same in both populations, all of a sudden you have enough data to maybe do a good job. And that will help you estimate your treatment effect. So those are three possibilities. If anybody has any thoughts about what exactly is going on, I'd love to hear. That's where we're at now. When I started this stuff, it was motivated by how to combine a bunch of genomic data. Now I have a whole lot more to combine. I'm working with a group at Illinois, which looks at social behavior in animals. We have genotype, we have gene expression, we have single cell gene expression. We're going to get spatial transcriptomic data. And the idea is similar. Can we combine all of these things together to understand how individual variation at the genotypic level results in behavioral variation at the phenotypic level? So I'm thinking that some of these ideas may help. So that's it. I hope I didn't run over time. If anybody has any suggestions or things to talk about, I'd love to. Thanks.
Thanks very much, Dave. It's a wonderful talk. It's very clear. And I think especially for the students who haven't heard of mediation analysis, this is a very nice introduction in the beginning.
I hope so. Yeah.
I hope that we can revisit the topic about the question I raised about this connection with TWAS. So what's your perspective about the TWAS?
So my understanding of TWAS is that they have their main interest is in what genes affect your outcome.
Right.
And their data structure has one data set where you have genotype data and gene expression data, like a EQTL data set like GTEx or something.
Yeah.
And then they have another data set where you have a GWAS, where you only have genotype and phenotype. And my understanding is the purpose of TWAS is, with that data structure, I still want to have this association. And so what TWAS does, correct me if I'm wrong, but what TWAS does is they learn this relationship.
Right.
Then in this data set, they predict G hat using that learned relationship and associate G hat with Y. And so ultimately, again, they're just focused on this arrow here, but I'm focused on this arrow, or this overall association.
Yeah, so my understanding is that a little difference between our understanding is that I think in TWAS, they didn't predict Y hat. They used the observed Y. So in other words, when they learn the relationship between G and Y, the data points or the observations are the individuals that have that have S and Y, but may not have G. Right. So they actually use G hat, which were predicted from the GTAC data where you have both S and G. That's something I understand. But I thought that they are still trying to, so even though they try to learn the association between G and Y, but I think the ultimate goal is that they're kind of trying to impute data for this triangle. So in this triangle, the GWAS data only give you observations of paired observations of S and Y, but not G. They basically imputed G for those individuals. And then essentially, given that imputed G hat, you could do what you have here. So this may not be called TWAS, but I feel like that's the ultimate goal of this genetic analysis. You want to have genotype, gene expression, and phenotype, right, the outcome. So ultimately, from mechanistic point of view, you want to know the association between S and Y. So you can easily use, say, gene editing to change your SNP. Hopefully, then you can alter the outcome. But on the other hand, it's good to know what gene is really functioning in the middle. that carries the effect from the SNP to phenotype. That's also my understanding, and I'm not completely sure if it's correct or wrong. So that's why I'm opening it up for discussion.
Yeah, I think I agree with what you said. They impute G hat, whereas and then their interest is G hat on Y. And again, my interest is Y on S overall. However, I think this imputation idea can also be carried over here. It's just that the data structure is a little different. So in TWAS, they have this data set and this data set.
Right.
But in my case, what you could do is you have one data set with EQTL and another data set which is just pure gene expression profiling.
Yeah.
And then what you could do is learn this, in my notation here, learn alpha hat from this data set.
Yeah.
And then impute Y hat on this data set and then learn that relationship. So if you have transportability of models and so on, then you can do a GWAS even without genotype and gene expression data, genotype phenotype data. So I think,
go ahead.
Yeah, I mean, I think in terms of imputation, that's where the connection is. I think in terms of conceptual problem, again, their focus is on a different association, and also I don't think their focus is on increasing power or anything. It's sort of imputing. I mean, it's just like imputing.
They try to explain some of the SNPs that would be found to be associated with the outcome from GWAS. I think that's the purpose. They try to find the genes that can also explain the phenotype, and then see whether those genes are close to those SNPs they cannot explain, and then try to make some story out of it. But back to your story, I think a related but also very interesting question is, if some of the variables are, like you said, the observations are imputed, how will the power analysis change? So suppose that your G is G hat, and you still carry on the mediation analysis. Will the power increase? And let's imagine there's no direct effect, just an indirect effect. Will there still be some power increase?
Yeah, I think so. I think so because a lot of the stuff, I mean, let's maybe take this easy situation here. This can be, the analysis of this part can be separated from the analysis of this part.
From this part.
Yeah, and I think some of, I haven't really thought that hard about it, but I think that independence between the G in this box and the G in this box may actually make things easier.
Okay.
But the fundamental power gain, I think, comes from these three ideas. And I mean, I don't know which one you want to think about. Let's just think about this one. Fundamentally, you're still making this assumption when you try to use this imputed G to get the overall effect. You're still making this assumption. So my guess is the power will still be stronger.
I see. And this assumption is actually, if you translate to that inequality, it's basically saying that either rho YG square or rho GS square is 1.
No, it just says, so this is always true.
Yeah.
It just says that this is also true. I mean, this doesn't say anything about what rho YG and rho GS have to equal. It just says their product equals the thing I care about.
Right, now, but then it's just that on the left-hand side, the plus minus term must be 0, so it must be equal, right? It can be equal.
No, it just says that. So, for example, I can give you a correlation matrix.
Okay.
Where these are equal, and this inequality is satisfied. So this, I guess what I'm saying is, this doesn't imply this. But if this is true, this can still be true.
I have a question.
Yes.
When you consider both indirect and direct,
yes.
Do you have a slide on that? And you say you still focus on the product.
Yes.
I don't understand why. Because here your goal is not estimation or not detection of association through certain paths.
Right.
Your goal is to detect overall association.
Right.
So why do you focus on a certain path?
Right. The reason is because we were so interested in gaining power, we wanted to use this extra information somehow. And this extra information is related to, our model says, this extra information is related to the information we already have via this. And so if we wanted to use G, basically we want to use this boxed equation somehow. So in other words, we thought about, well, what if we can, instead of regressing Y directly on S, that would just use G, what if we could capture information about Y in G? And then because we have G, we measure G, we can fit this equation here and use it. And so that's the motivation behind this statistic here. So it's not saying that we want to go through that path necessarily. It's not saying that like we don't care about that path. It says that that's the best way we could think of for how to use the extra G information that we had. And then, I mean, I went over this not very well, so probably didn't come across, but we did also consider interpolating between the usual OLS estimator for just ignore G, just go Y on S, no path, nothing. And then this C equals 1 stands for just use that projected test statistic. And we considered convex combinations of those. And so in some cases, it is true that focusing on the information through G doesn't really help you too much. For example, here, if you just focus on the information about Y captured through G, you have lower power. And in some cases, it is beneficial to, borrow some from the usual way to do things. Does that make sense? Did that answer your question?
Sort of. Yeah. No, I don't have an answer myself, so I just curious. Why
Yes, I have to say that we tried other things, but couldn't come up with a better way. And so there may exist a better way. We couldn't come up with one. So intuitively, we just said, if we want to take advantage of this structure, then we want to project this onto this so that we can take advantage of this relationship here.
Okay, without thinking of it, so naively, I would just start with a two-dimensional, 2 degrees freedom test on the first regression.
Yeah.
I know it's 2 degrees freedom, so may lose power because of that, but
yes,
that would consider both G and S.
So if it was 6 years ago, but in this paper's supplement, I think I tried that, and you don't gain power.
Okay.
And I don't remember why, but you don't gain power. Yes.
Thank you.
Yes, thank you.
I also have a question.
Yes.
Yeah, with regard to this, when I look at the estimation of G, like we regress S, like G is estimated from S, or rather it regressed from S. Does that mean that all the variables in G, because G is high dimensional as stated there, does that mean that all the variables in G are instrumented, whereby S is technically the same dimension as G? And if not, then if there are some variables in G that have not yet been instrumented, are we not controlling for them? Because I don't see in the two-stage linear regression or the instrument variable method, we usually control for the variables that are not instrumented. So in this case of yours, why are you not controlling for these variables that are not instrumented?
So I will try to answer your question, although I have to say my instrumental variable knowledge is not very good. But I think what you're asking is, not all of the G affect Y through S. I mean, sorry, not all of the G's are involved in this relationship, basically, right?
What I mean is that S kind of might only affect part of G, not all of it.
Yes. So this model tells you how much of which G are affected by S. This model tells you which of those G affect Y. And so this quantifies how many G that affect Y are affected by S. And we allow, zeros here, like not all the G have to be affected by S. Not all the ones affected by S have to affect Y. And that's fine.
But does that not mean that there's some effect that you've not accounted for? Because technically there's some parts of G that affects Y, but it's not affected from S. So are you not supposed to take, like, to control for this part of G that is not affected from S, or is not affected from S but affects Y? So are you not taking that into consideration?
I think we are, just because they're in here. Those G that affect Y are all captured by this alpha 0 term, regardless of whether or not those G are also affected by S. So if you have, let's say if G1, G2, and G3 affect Y, but only G1 is affected by S, that's okay. This beta 0 will only contain the effect through G1. And so I guess if you're asking about, do we control for the other G, I would say yes, because they're here. I guess I should also say, if you want to add covariates, you could, and I didn't talk about that, but you can add additional covariates as well. But yeah, I mean, I would say some of the G that aren't affected by S are still captured by non-zero components of alpha 0.
So for example, if I were to run this model in R, for example, I'm just saying, can I be able to use the IVReg, for example, package, that is the instrument variable package, whereby I have to control for both the instrumented and non-instrumented variables? Because in that case, I feel like the way the model is written here does not fall in the consideration whereby we are talking of instrument variables. So probably this is kind of different from the instrument, the tackling of this is kind of different from the way instrument variables is tackled.
Yeah, so I think that may be the core issue. Again, I'm not familiar with instrumental variables too much, but their focus is finding this arrow. So yes, it is indeed important for that literature to figure out which G are affected by S and which are not, and you only care about the ones that are affected through S. So because I'm not focused on this arrow, I'm focused on the overall relationship. I think that might be a reason why things are different. I don't know whether you can run it through IVReg, but we have you can download our package on R and on GitHub, I guess, and try it. I think the settings are different enough that I think your concern about controlling for G's not affected by S is okay here.
Mm-hmm.
But I don't know, maybe, yeah, I would love to talk more about this. I don't know 100% the connection between this and IV regression, but I think we're okay here.
Thank you.
Thanks.
Yeah, I think one challenge to me, at least about the field, is that there are so many terms and jargons that are used, right? Especially there are mediators. I think in causal inference, you also have another term called moderator. So I think to me, those terms need to be memorized. So you can actually, if you don't work in the field, you have to refresh your mind every time you see those terms. Otherwise, they don't actually directly translate into mathematical definitions.
Yeah, I should say that there's a whole causal field for mediation that I'm not working on. So like you said, there's moderators, there's natural indirect effects, there's other types of indirect effects. So there's all these definitions. Things change when you're nonlinear. I'm just trying to write down these easy regression models where it's easy to identify mathematically, what parameter you're talking about, and then work from there.
Yeah. Actually, I have a conceptual question regarding the high-dimensional mediation analysis with still direct effect. So my feeling is, at least in this genetics context, if you include all genes into your G, that becomes high-dimensional. Then how could there still be direct effect from S to Y?
Yeah. So, if S has a coding. If S is a coding variation, it may not affect the expression level of G, it may affect the structure of the protein, and that can affect Y, right? So hemophilia, the mutation affects the kink in like the whatever beta globulin.
Oh, I see. Yeah, because G here means expression only.
Yeah.
Okay, I see. Then that makes it. Yeah.
Our hope is indeed that the most of the SNPs that people study, right, probably regulate transcription instead of regulate protein structure or something. So our hope is that most of the S's that you would use this for are gonna proceed through gene expression.
I see.
Yeah.
I see.
Can I ask, related to kind of the power anomalies in mediation paper, the Judd and Kenny paper, they also talk about kind of a different anomaly where we're taking the product of two coefficients and the power differs depending on which one is which number. So the indirect effect could be 1, and if one coefficient is 2 and the other is 0.5, the power differs depending on which one is 2 and which one is 0.5. Do you feel like these results have anything to say about why that happens? Or is that something completely different?
So the results that we worked on don't have anything to say about that because we just focused on the product. However, we certainly noticed that because when you actually like simulate these data or work with these data, the what number alpha is and what number B0 is, right, depends on this. And I think this is exactly what you're saying. You can swap some values between alpha 0 and B0 and get the same value for beta 0. And those do end up affecting, I guess, finite sample power. We didn't study that too much. And I think also we were looking at asymptotic results. So we don't have a whole lot to say about finite sample. The one thing that we did discover is if you, for example, let's take B. So suppose beta 0 is large, right? Should be easy to detect. However, suppose that's because alpha 0 is small and B0 is big. If that's true, then a lot of these G will be associated with each other because they all stem from the same, let's say S is low dimensional, right? So they all stem from the same small set of S's. And so the correlation structure of the G will be very correlated because they all come from a lot of the same S's. If that's the case, then you have high multicollinearity here. And so in finite samples, even with like lasso or whatever, you'll still do a poor job. I don't know if that's exactly what Kenny and Judd were talking about, but that's how we found it manifested in this work, if that relates at all.
Yeah, I think that's very in line with what they talked about and what other people talk about.
Okay. Yeah, this is much more complicated because you have, right, two different things that can give you the same result. So that took a while for us to realize. Yeah, thanks for the question. I'll go back and read that paper and that section. Oh yeah, and I think this paper may talk about, this other one may talk about it. I'm not totally sure.
And so Dave, there's a question from the chat box from Gabriel. Maybe Gabriel, you can read it out. Oh, sorry, I think we cannot hear you, but that's okay. So Dave, can you see the text in the chat box?
I just, because as you know, for full screen, I can't see the chat box. I just saw everybody's chat, so sorry if I missed them. Can you?
No problem. Actually, I can read it out.
I see it. Can you elaborate on the high dimensional causal inference paper you mentioned in the discussion? So I don't, okay, so the question is, basically, we have to have the right arrows in the right directions. And we don't know how to do that. And I don't know much about the literature for how to do that, but I just know that Bielman and his colleagues have worked on this type of idea before. So I have not thought about how to use their method to get the right DAG structure here. If you have any thoughts, that would be ideal.
Okay, so that becomes a selection of mediator problem.
Yes.
Yeah.
The mediator selection and direction problem.
Direction problem. I see.
Yeah.
And okay, I see.
Sorry, I know that wasn't a good answer. I don't know much about how they actually go about selecting their things.
Yeah, actually, so I have another practical question to ask. So in your collaboration with your collaborators who generated S, G, and Y for the same set of objects, so then what's your suggestion for them? Do you recommend they do the mediation analysis or not to capture the association between S and Y?
Yeah, so that is a fascinating question. Right now, I don't recommend they do it I don't recommend they do it for anything other than exploratory analysis right now. And that's because, at least for actually what this paper talked about, this paper here, for the stuff that I've described, you need a lot of extra assumptions. If you want to gain power, you have, in the standard case, all you have to assume is that Y is related to S somehow. If you want to gain power, you have to assume how Y is related to G and how G is related to S. And so you can run into problems there. And also, like Gabriel was talking about, wrong mediators, the wrong directions, and so on. However, I would recommend it if you can't do anything else. And so what we're trying to do now is understand how genotype affects behavior, social behavior in animals.
Right.
That's very, and we can't get 4 million, bees or anything like that, because, that's what Vischer needed for his height study, and there's no way we can get that, right? In that case, the best we can do is maybe get a small number of samples and just collect a lot of other data types. So bulk expression, maybe attack-seq, maybe, spatial, single cell. If you have all of that and you just want to get to some set of SNPs, there's very low chance that if you just do a standard GWAS, you'll find anything, given the variability in the phenotype and the low sample size. And so there, I would just say, yeah, try it. Why not?
Okay.
Yeah.
I see.
But I mean, that's why I want to make this actually work. For example, the last thing I was talking about, this non-parametric F, if you can find a non-parametric way of estimating F, that would be more realistic. That's what Rachel's working on right now. And at some point, I hope this will be usable.
I see. So for this one, it said that it's from your collaboration about the surrogate marker study, right? So from that part, but you said that this also applies to the mediation analysis.
So it's the exact same math. So surrogate markers and mediation are identical. And we didn't realize that before. But if you look at this picture over here, it's exactly a mediation model, except that
and here the mediator is known for sure, right? Here the mediator doesn't need to be selected.
So yes, so the surrogate marker literature assumes you have a good surrogate. And Ross Prentice has all of these definitions for what a good surrogate needs to satisfy. So they usually, I think a lot of the literature will assume that a lot of those criteria are met. And one of the things Layla was trying to do is figure out, if you don't know that this is a good surrogate yet, how could you find out? And one way to find out is, can you estimate what proportion of this effect on this is actually going through that surrogate? Because it better be high. If it's not high, it's not a good surrogate.
That makes sense.
If you look at the math, for example, if you look at the assumptions in this paper, they're like identical to the mediation assumptions, and they don't really, I guess people haven't put these together yet, it appears.
Because I think it's just because their origins are kind of different, right? The mediators were some variables you don't choose, you just observe. But this, I think that's kind of more like observational data in mediation analysis. But this one, you are kind of more like in a clinical trial or more like experimental design. Yeah.
Exactly. So the data generating structure is different, but the conceptual issues are really close.
Okay. Yeah, I see. And in that last figure, when you have that non-parametric estimation figure, that one here, the Y is binary, right? Or no, Y is not binary, but the treatment is binary.
Yes.
Yeah, intervention. Yeah.
Yes. Yes. And also thanks to Amanda and Gabriel for your paper and also the elaboration on the Buellman paper. Appreciate it.
I think the overlap between the surrogate marker and I would say like the psychology mediation literature, you can find it if you start looking at the effect size literature for mediation analysis, because that's really where, people have focused in the surrogate markers on kind of proportion of variance explained, whereas in psychology, a lot of the time the focus is on more like standardized coefficients. And so people who've brought together kind of what are the possible effect sizes to use have pulled from both the surrogate marker literature and mediation overall.
Okay, great. Yeah, I haven't explored that literature, but I think Rachel was on the call now. So Rachel, you got work to do.
So Amanda, I have a follow-up question from what you said. So what are the possible effect sizes they have considered in surrogate to quantify the effect of a surrogate marker, or how good a marker is?
So in the surrogate marker literature, most of the focus is on, because they're dealing with these cases where, like Dave said, consistent mediation, where the direct effect and the indirect effect are of the same sign, then you can focus on what proportion of the total effect the indirect effect is. And if that proportion is large, then it's a good surrogate marker.
That's what Dave just said, right? In that paper you showed.
Exactly.
Yeah, okay.
So that's been the focus in surrogate markers and kind of the more medical literature. In psychology, people are more often running into cases where the indirect and direct effect are of opposite signs, or they don't know what sign they're going to be. And so those types of proportion measures can be quite misleading, either because the total effect is near zero, but the indirect effect is large. And so you can get the proportion being, you know, huge for kind of no reason, more because the coefficients are of opposite signs. So the focus there is a measure called kappa squared that came out, but then it was shown to be wrong, and then there was a new version that came out, and it's trying to get more at kind of a variance explained type of measure, like an R squared, but combining the information across the two models, which is what's then quite difficult to do.
If you can send like a reference to maybe a paper that surveys some of this stuff, that'd be really helpful.
Yeah, happy to.
Yeah, thanks. I was about to write down kappa squared, but then you said it was wrong, so
yeah.
I'm not gonna write it down anymore.
The effect size literature in mediation analysis is quite fraught.
Okay.
It's kappa squared was the only one that was kind of mathematically wrong, but it's very difficult to recommend an effect size because it often depends on whether you have consistent or inconsistent mediation, what you should be using. And oftentimes someone doesn't know ahead of time what they're going to have. So the seeking out an effect size that works in all cases is still kind of an open question. Yeah.
Well, yeah, it's cool because I guess there's a lot more work to be done conceptually and maybe mathematically too.
Yeah, absolutely. I think this is great. So I'm from the psych department, and I do research and mediation analysis, if that's not obvious. But thank you so much for this talk. It was really, really wonderful. It's very cool to see this applied in kind of a genomics, genetics situation. That's something I'm totally unfamiliar with. So.
Well, yeah, thanks for your comments, and thanks everybody for your comments. I'm totally gonna look up all these things in the chat box, so really appreciate it.
Thank you so much.
Yeah, thank you.
Thank you.
Jessica, thanks for inviting me. It's been really fun.
My pleasure. It's a very wonderful talk again. Yeah, so Dave, I can save the chat. So yeah, I did it, so I can send it to you. Yeah.
Okay.
Okay. All right, so thanks Dave again, and thanks everyone for your attendance. Yeah, we look forward to more discussion. Bye.